The broadcast is now starting. All attendees are in listen-only mode.
Welcome everyone to our webinar today. My name is Shauna White and I'm the Corporate Marketing Manager at DNA Genotech. We're pleased to have registrants from around the globe here with us today. But before we begin the webinar, I have a few items to review with you. Today's webinar is being recorded. All participants are currently muted. We will be allowing time for questions and answers at the end of the webinar. Please type your questions using the questions window on your GoToMeeting console. We will queue your questions and respond to them at the end of today's webinar. If we do not have time for all the questions, we will respond to you individually after the webinar is completed. Thank you again for joining us today. I'll now turn the controls over to John Chatburn to get our presentation started.
Excellent. Thank you very much, Shauna. And thank you all for joining us today for our webinar entitled Overcoming Challenges in DNA Sample Acquisition for Cancer Research. It's quite timely and unique that we have this technology in lieu of the fact that we have a volcano spewing ash into our atmosphere and preventing travel. So it's great that we can all be together here today, at least over cyberspace. Okay, to talk a little bit about the agenda and what we're going to speak about today. First, we'll talk a little bit about who DNA Genotech is and what our company does. We'll spend some time putting some framework around the challenges that cancer researchers face. We'll then talk a little bit about how we can overcome some of those challenges with Origin DNA. I'll pass it over to my colleague Aaron Del Duca after that, and he will take you through how Origin DNA is being used within publications and helping to enable discovery. And then finally, we'll follow up with some questions and answers after that. Okay, so who is DNA Genotech? DNA Genotech is our company name. We are located in Ottawa, Canada, which is located somewhere beneath that red maple leaf you see on your screen. As a company, all we do is work with biospecimen collection, transport, storage, or purification for molecular analysis and testing. Our company was founded by a man named Dr. Hein Birnbaum. And Dr. Birnbaum was a cancer researcher at the Ottawa Regional Cancer Center. And uniquely enough, his research and some of his inventions for his research became our lead products that we're going to talk about today. The company has been around for about 7 years now. And since that time, we've been used now in about 4,000 labs in 85 countries worldwide from right here in Ottawa, Canada. Some of the customers that we work with are listed here on your screen. You can see we work with a variety of different groups from academic institutions that are doing basic medical genetic research and even cancer research. As you can see, we have MD Anderson Cancer Center and Cancer Research UK. Our products are also used in hospitals for clinical trials and genetic screening programs. And they're also used by commercial entities for diagnostic development and even pharmacogenomics and personalized genomic companies now. As you can see, Navigenics and 23andMe listed as well. Okay, we're going to move on now to talk a little bit about some of the challenges that cancer researchers face. And of course, some of these challenges are ubiquitous amongst all researchers, but there are some that are specific to cancer research itself. There are a wide variety of lines of cancer. There are a wide variety of cancer research studies that are ongoing, and it's particularly looking at the variety of etiologies behind different types of cancers. There are some problems and challenges that are being seen amongst all types of research. There's been a lot of focus in recent years looking at gene-environment interactions and trying to understand what the lower level risks are for identifying genetic causes of cancer. There's also been an increased interest in looking at the effects of subgroups or effect modification, those smaller processes that occur within the groups that are being studied. And because there's such a wide variety of cancers out there and there's such a wide variety of expertise required, the teams that are studying them become larger. You need more interdisciplinary study teams, PIs that have a diverse background to help contribute to these studies. Of course, the studies are getting larger and larger with more diverse groups and populations required for the study's design itself. Of course, the technologies that are being developed recently for the analysis of genetic material are becoming larger, more expensive, and certainly more sophisticated. All this leads to increasing budgets and increasing the size of study, which of course overall will increase the costs. There are some very practical challenges that are faced when conducting research, and specifically cancer cohort research itself. Large studies often require a large number of samples, and in terms of DNA samples, what you need when you collect DNA is a very, very high quality genomic sample. You require to collect from a large number of people. Oftentimes it's case control studies. Blood will be often difficult to obtain, and blood is what is typically used in these types of studies, but you can create compliance problems when looking at collecting blood versus another sample type. Blood is often not possible. In fact, there's large study populations going on now where it's remote population, difficult to access, difficult to attract people to come into centers to provide blood, so it may not be possible to actually achieve. And of course, shipping and processing blood is difficult. If you're sending directly to a main center and you're trying to achieve a sample from a remote location, it's very costly and difficult to ship to the main study center. The question really is there an alternative to blood, which is the established method that would perform as well as blood and remove the challenges that are faced surrounding using blood as that collection methodology? We believe we have the answer, and that is Origin DNA. For those of you who may not be familiar with what Origin is a means of collecting, stabilizing, transporting, and then processing your DNA sample, preparing it for its downstream application, all in one. It's a non-invasive self-collection device, meaning that it's provided to the donor without the oversight of a technologist or phlebotomist for delivery of the sample. What I have and pointing here to on the left of the screen is what the contents of our Origin kits contain. There's a plastic shell, and inside that kit comes our leaflet and the collection tube itself. The larger collection tube here, shown in the picture, inside the top of that lid is a reagent that when the cap is closed is released, and it mixes with the saliva that is given by the donor, and that stabilizes the DNA within the saliva at room temperature. You would then remove that funnel, place on the accessory cap, and that's your sample stable at room temperature inside this little tube. Below the instructions that we're showing here are the actual instructions that come in the inside lid of the collection kit. So it's literally as easy as putting saliva in the tube, closing the tube, and then changing the cap, and you're done. On this next slide here, I've got what we call our DNA life cycle. For those of you who work with DNA, you realize that there are more to working with DNA than just collection or just using it for your downstream application. There's multiple components to how we collect our samples, how we work with them or store them, and then how we get the DNA out of the sample and use it for our downstream application. What we're going to walk you through now is what we call our Origin DNA 100% solution. It's how we use Origin and integrate Origin into this system to provide a solution that covers all aspects of a DNA life cycle, from collection to stabilization and transport to the purification protocols, and then finally for use on your downstream application. First, let's talk a little bit about collection. The first thing that we should touch on regarding moving from an invasive methodology for collection like blood over to a non-invasive methodology like saliva is patient compliance. If you move to something that is non-invasive, you will immediately get an increase in your donor compliance or the willingness to provide a sample for a study. The study title that you see here is from customers of ours at the Karolinska Institute. They came to us a few years back and wanted to conduct a study, a large cancer study looking at, I believe, prostate cancer in Swedish men. And men in general are a notoriously difficult sample set to get from, or a population to achieve samples from. They just seem to be unwilling to provide samples. So what they were looking for was an easy way and a non-invasive way to collect samples and to get these shipped through the post in a very easy way. What they did was run a pilot and sent out 611 samples to men via the post. They got 490 samples returned via the post. 93% of which arrived in the first 3 weeks. That is a huge number of samples coming back very quickly, and the reason for that is because when they eliminated the need for a phlebotomist and a center to take blood, they were able to then move from what we call a serial process, or sampling one at a time, to a parallel process via shipping out multiple samples simultaneously, and essentially speeding up the collection process and shortening the study duration overall. The most unique aspect of this particular pilot that they found is that they had almost 100% increase in the number of donors that would provide a sample if offered a non-invasive method compared with blood, which is an invasive method. So you can see that they got 80% compliance rates when they used Oragene for the study. Following this study, they went on to use Oragene for their larger cohort and have finished now and have upwards collected 40,000 samples. And Karolinska continues to use Oragene for some of their larger studies, and of course their biobanking as well. So getting your sample to and fro from your donors is one aspect, of course, and compliance is a big part of that. You need to make sure that you have a very, very high yield when you're doing research studies because of the variety of downstream applications that you may or may not be using. The chart that I have here is showing the variety of collection methodologies that exist right now. And of course, Oragene, you can see, performs very, very well. In fact, it's got some of the highest DNA yields at an average of 110 micrograms. We have to note that there is variability within that sample type. Just due to the biology of saliva, there is a variability in the number of cells that yield DNA within saliva, and so there will be some variability in that sample, but on average it's a very, very high yield. Next to it is venous blood, a very consistent yield at around 80 micrograms, and then mouthwash at around 36 or so, and then buccal swabs at very, very low microgram quantities. The yellow portion of the graph represents the bacterial contamination, or the portion of that sample that is from bacterial origin. And with Oragene, it keeps the bacterial content very low. And the reason for that is that although there is bacteria in your mouth that will be deposited into the tube itself when you deliver saliva, the stabilization medium in Oragene prevents growth and kills the bacteria on contact. So there's no proliferation of bacteria within that sample, and therefore you have a very low bacterial content to that DNA sample. Blood, venous blood, there is essentially nil. Mouthwash, almost 40 to 50% of that sample will be bacterial contamination. The reason for that is that mouthwash is designed to rid the mouth of bacteria. You spit that back into your sample and you've got a nice bacterial sample there. And of course buccal swabs is extremely high as well. And the reason for that is that bacteria tend to congregate around the teeth and gum line where swabs, the method for swabs gets the DNA from. And there is no stabilization medium for a buccal swab, so bacteria tends to grow on the swab itself, and you end up with a nice high bacterial content. One note to point out, the bullet here where we explain that 50 to 70% of the cellular content of saliva is white blood cells. We've had a lot of controversy over the years as to whether or not saliva and blood would be a compatible source of DNA, or whether or not you can use saliva in place of blood. From some of the research that we have seen, and some of these sources at the bottom, that 50 to 7% of the cellular content that yields DNA in saliva comes from white blood cells. So it is the same source as that is blood. Okay, so if you have a nice high yield DNA sample, that's fantastic. If your DNA sample is degraded, doesn't matter how much DNA you have, it's going to be useless for your downstream applications. Probably one of the most interesting things about Oragene is its ability to yield very high quality DNA as well. On average, the molecular weight fragments that come out of Oragene are upwards of 23 kb in size, and sometimes larger, 40 to 50 kb. So if you're running high fidelity downstream analysis techniques or long range PCR, something that you need large fragments, it's not going to be a problem. The purification process as well for Oragene yields very, very high quality DNA in terms of its adsorbance ratios, which sit at around 1.6 to 1.9, which is about the same as blood. So you make sure that there's no PCR inhibitors, and the removal of any contamination is done through the purification process. Okay, moving on from collection now to stabilization, transport, and storage of your genetic sample. Probably the most valuable and most brilliant part of Origene's design is its ability to stabilize DNA at ambient temperature. I suggested that we get back high molecular weight DNA at 23 kb, very, very large fragments. So what I have here is on the top portion is a gel from our accelerated aging studies. What we did was take samples and we stored them, Origene saliva samples, and stored them at different temperatures. You see 24, 37 degrees Celsius, and 50 degrees Celsius for a varying number of days, ranging from 0 to 187 days at the longest, just to determine whether or not, under these conditions and under a certain time duration, if the DNA would break down. As you can see, we took the samples out, ran them on a gel, and you're still getting the majority of the sample, or the DNA, being maintained at 23 kb sizes. This indicating that the DNA is not breaking down. Even after 187 days, nearly 6 months, at 50 degrees Celsius, the DNA is not degrading. The lower gel is showing our real-time studies in which we took samples again, Origene saliva samples, and this time we put them on the shelf for a period of 5 years, and then took the samples back off, purified them, and ran the DNA on a gel to determine if there was degradation over that time duration. And as you can see again, we have very clear bands forming at 23 kb, indicating that we have very high quality DNA still being maintained after 5 years at room temperature. And this, of course, the ability to stabilize DNA at ambient temperature allows for field collection or transport in the post, because whatever conditions it may encounter while in transit won't be a problem. And that's including freezing as well. You can freeze thaw these Origene samples many times over without the fear of shearing or degradation of the DNA, which of course then makes it ideal for shipping through the post, because if it encounters cold temperatures at altitude, it isn't a problem whatsoever. Okay, so on to sample transport. So getting your sample to and from your donor. The ability to stabilize DNA at ambient temperature makes this possible. But there are some regulations shipping biosamples through the post. The great thing about Origene is that saliva is considered an exempt human specimen, meaning it falls into the most lax category that IATA classifies for a sample. And all you require to ship a sample in the post, or an Origene sample in the post, is a biohazard or biosample baggie that I'm circling here. That's the minimal packaging required. The other components that you see here are a postal package or an accessory that as a company we can provide so that you can ship your samples to and from your donors. And inside this package there are instructions indicating to the donor how to not only provide the sample, but then how to assemble the package and ship back to donor so that you ensure that the sample gets to them and back to the lab as safely as possible. I should also note at the bottom here we have some certifications. Our company is an ISO 13485 registered company, meaning we have a quality management system for the production of medical devices. And the kit itself is certified CE as an IVD in European Union. So it means that for shipping over the post or using within labs or shipping over borders that our kits comply with the local regulations. Okay, now moving from transport and storage on to DNA purification, or how you're going to get your DNA out of your saliva sample itself. We provide our own purification chemistry and protocol with the kit, which we'll talk about in a moment, but Origene is also compatible with a variety of third-party chemistries that exist on the market. If we haven't encountered them, we will work with that company to ensure that the chemistry does function with our kits, but we also have a number of protocols already developed. As I mentioned, we have a chemistry that we provide with the kit that allows for the extraction of the DNA from the sample itself. This is an ethanol precipitation protocol. It's very easy to follow. It takes about 30 minutes to do. I've done it myself, and if I can do it, it means anyone could do it. It's a non-hazardous protocol. It's not chloroform-based, so it makes it very easy to use and simple to work with in the lab. We do have many clients who have throughputs in their labs that range from, say, very low throughput manual purification protocols to extremely high throughput, where they're doing thousands of samples at a time. We work with a variety of companies to ensure that our Origin kits are compatible with their automation platforms. And so I have some of them listed here with the range and throughputs that they can take. If there's a company that you work with or you have a robot in your lab that we're unfamiliar with, again, we work with the company to ensure that we can optimize it for the laboratory workflow and that you can get the results that you would like to see out of your robots. Okay, so on from purification now to talk a little bit about the variety of downstream applications that Origin is compatible with. I will note that there are many, too many to talk to individually. However, just to indicate going the comparability from saliva to blood, Origin is compatible with just about everything out there, and the reason is because the same source of DNA that you would get from blood. So anything you can use blood on in terms of the DNA, you can use Origin on as well. What we have here is listed some of the typical and most popular downstream analysis techniques that exist right now. Again, we have protocols that are optimized for integrating Origin samples onto these systems. If there is a system that we're unfamiliar with, we will work with the company to design a protocol to make sure that it is compatible and it works. A special note I'd like to make at the bottom here, the DNA Genetic Partner Program. We've initiated this over the last year to partner with different laboratories and technology providers to ensure that we can provide Origin-approved labs and technology vendors. So if you're looking or designing a project or have questions about which technologies and which laboratories are compatible with us, we can provide that list to you and connect you with someone who knows how to use our products and is very familiar with them. Okay, at this point, I'm going to pass the controls back to Shauna, and you will be hearing shortly from my colleague, Aaron Del Duca.
All right, Aaron, I think I've got controls passed over to you. You should be all set to go.
Thanks very much, Shauna. Okay, we'll just make sure we're going. There we are. Okay, so far in the presentation we've had a chance to learn about who DNA Genotech is and how our products add value throughout the life cycle of a diagnostic specimen. And as John alluded to, after having the Origin product line out on the market for about the last 6 years or so, we've had the very good fortune of wide adoption. I think over 85 countries, mainly due to our ability to improve compliance rates and participation in genetic research programs. And as such, we've been a part of about almost 200 peer-reviewed publications covering very diverse subject matter from basic genomic science through to personalized medicine, pharmacogenomics, and even molecular diagnostics. So I'll switch gears a little bit and I'll summarize a few key publications where Origin has played an important role in the investigator's ability to either carry out the study at all or where the sampling technique has had a significant positive effect on assay performance and data analysis. So before I click on, I should mention that the publications that you're seeing here is just a really short list, and the link at the bottom of the slide here will take you to the full list. And if you have any other questions or if you'd like to dive deeper into a topic, I'd encourage you to check out that really valuable resource. Okay, so let's talk a bit about enabling discovery with Origin DNA. The first paper I'll talk about is by Liying Zhang and Kenneth Offit and colleagues at the Memorial Sloan Kettering Cancer Center in New York. This was published in the Journal of Molecular Diagnostics in May 2009. And it's a particularly timely topic given that Myriad Genetics and the ACLU are litigating over validity of BRCA gene patents, and I'm sure that patent law is very invigorating, but I'll park my opinion on that and just stick to rapid and reliable testing methods for these mutations. So in very general terms, the authors are exploring what it would take to improve the standard of care when it comes to screening for BRCA1 and 2 founder mutations in women of Ashkenazi Jewish descent. And in their view, women newly diagnosed need much faster turnaround times than are currently available in order to take all that counseling information in, consult with loved ones, and make informed decisions. So you can imagine, one of the challenges here is that when counseling women in this demographic, that you'd have a need to sample further and wider than New York City to really understand the pedigree. So you can envision, a patient at Memorial Sloan Kettering who's got an aunt somewhere in Chicago maybe, and her grandmother is in the Middle East. So being able to collect from those individuals would really help sort out the hereditary pattern in the family, and that ultimately will be very valuable to the clinician when he or she is trying to assign some level of risk to the patient seeking counseling. So in this study, the authors took a two-pronged approach to overcoming these challenges. They first were hypothesizing that improving the availability of high-quality samples would speed up the process. So in addition to taking blood, they looked at broadening their sampling methods. They turned to Origene for collecting saliva samples, and they also tried to access archived pathological tissues of relatives who had undergone surgical procedures sometime in the past. And they found some pretty interesting results. Firstly, they found FFPE tissues, when available, to be helpful, but it posed challenges to their traditional methods, which included Sanger sequencing or PCR, RFLP mutation screening methods. And as you all know, formalin-fixed, paraffin-embedded samples preserve morphology, which is great for the pathologists who are ordering them and preparing them, but they really hang a beating on the nucleic acids in those samples, making it really difficult to get them to work for molecular diagnostics. So to get around the problem of degraded DNA in these samples, the authors came up with a new assay based on pyrosequencing using carefully designed, very short amplicons. And so this was a good start, but they found that these FFPE samples weren't really as available as they would like. So it hadn't fully resolved the problem of accessibility and speed and turnaround times for these tests. So that motivated them to introduce another sample type, Origene saliva collection. So that enabled them to, access relatives and who weren't within driving range to the clinic. So the first thing they did was send Origene kits to 30 patients who had previously received care at Memorial Sloan Kettering in order to compare the accuracy of their genotyping method as well as the quality of the Origene sample. And they found that not only did the samples work beautifully, but that offering a non-invasive sampling method could really promote the uptake of genetic testing. And in the end, they found that this method reduced the complete turnaround time to less than a week, creating a huge benefit for the clinicians, the patients, and their families. So if you're interested in learning a bit more about primer sets that these authors used or which mutations they were screening for, again, please check out the website and all that information will be available to you. So the next study I'd like to talk about concerns genetic susceptibility to childhood leukemias, and it was published by a long-time Origene user, Anand Chokalingam and Patricia Buffler at the University of California in Berkeley, and this was published in late 2008 in Radiation Protection Dosimetry. This is a bit more of a review article. So the advantage here is that they take a look at a very broad base of the literature, and they try and sort of distill down some of the best practices that they've gathered, as well as from their own experience in the Northern California childhood leukemia study, to sort of convey what they found to be their key hurdles and potential solutions to overcome them. One of the big challenges that they face is they've chosen to study acute lymphocytic leukemia, which is very rare. And as most cancers are, it's complex and heterogeneous, which is to say that many factors contribute to the disease, and those factors are low penetrant, so they contribute only a very small effect. And on top of that, gene-environment interactions can make it even more difficult to distinguish the inherited effects from conditions like air or soil quality or even the diet of the patient. So when you're tackling really complex subject matter, like many of our listeners are, there are many hypotheses that need to be tested. But the statistical power to show significance for any one of them is a huge challenge, and that requires very, very large sample sizes. And let's face it, it takes quite a lot of time and a ton of money to do these kinds of studies. So what can be done to overcome these challenges? Really briefly, Chokalingham and colleagues took the divide and conquer approach and formed the Childhood Leukemia International Consortium, or CLIC. And by teaming up with research-minded cancer specialists in 10 different countries, there were about 20 investigators in all that participated in their consortium. They really broadened the reach of the study and made it much easier to find cases that fit their very well-characterized phenotype. Also, the authors noted that switching from heel sticks to buccal swabs really helped, but when you're working in a consortium, not only do you need compliance, you also need enough genetic material to share as a resource among the group. So buccal swabs didn't really get them all the way there, so they then switched to Origene back in 2007, and that really helped them achieve the samples that they required. And it made, as John alluded to, shipping the samples around very, very easy, just using regular post. So they were collecting from 10 different countries and then sending them all back to one central processing facility in Berkeley. And it sounds like a simple thing, but when you're working with limited budgets, that can have a huge impact. So again, going from a serial process where one phlebotomist collects from one patient at a time to a parallel process where you can have a mass sample dissemination and then within a few weeks get them all back, that allows you to really take advantage of economies of scale and batch processing. And that can ultimately affect savings, which is what it's all about. So this diagram that I'm showing here is the model that Chokalingham and colleagues used to approach this really complex disease and test as many hypotheses as they possibly could. Some call it a fishing expedition. I call it sort of a hypothesis-free, unbiased approach to genetic discovery. And what they did was take a look at a really large set of SNP markers from about 800 to 1,000 cases, and then replicate that with similar sized groups to confirm that the linkage disequilibrium that they were seeing was actually real. So then they'd move from things like genome-wide association studies using microarrays down to more targeted real-time PCR-based methods, and then finally to sequencing and mapping to really ascertain which genes were presenting real effects with their phenotype of acute lymphocytic leukemia. Okay. So the next slide, which I'm trying to pull up here. Okay, is Spit for the Cure. And you may have heard for Run for the Cure. It's a charitable event that raises funds and awareness for breast cancer research. And we had a very unique opportunity to do something pretty special with Dr. Susan Cadlebar at the University of Arkansas. And it's pretty rare, but every once in a while, a social event, a charitable event like this will actually bring together a very desirable population from a genetic perspective. So in the photos, you're actually looking at thousands of breast cancer survivors, their families and friends and colleagues. And Dr. Cadlebar saw this as a really a good opportunity to reach out to the community and to sample from willing donors to participate in their study. And ultimately what they did, in conjunction with DNA Genotech through what we call the Helping Hands program, was we actually sent down volunteers from our company to the Spit for the Cure event, and then through through the annual event, in 2 years, we've been able to help them collect over 10,000 samples, and they're very well on their way to 40,000 to meet their statistical power goals for that particular case control study. So again, because it's a non-invasive method, it was actually very easy to do. On the right-hand side, you see women filling out their consent forms and getting ready to participate in the fun run. And at the same time, they're delivering their saliva samples. And that would have been inconceivable to do in pretty much any other way. So again, you're out there out in the open, could be warm, could be cold, doesn't matter. You're giving a saliva sample into a tube that maintains it at pretty much any condition. So that's something that we're very, very proud of. And they've gone on to actually patent the name Spit for the Cure. So expect to see some of these happening in your communities, wherever you might happen to be. So to summarize, this is definitely an easy way to collect, whether it be supervised in a clinic or unsupervised at some sort of charitable event. It can be done by the young and old, so it's very, very simple. Everything about the kit is non-toxic and non-hazardous. So if you're collecting from a child in their home, you don't have to worry about anything coming to harm your donors. It's reliable. It doesn't matter if you've never participated in a genetic study. It doesn't matter what demographic you're sampling from. We make it very, very easy, and the quality of the sample will definitely work in whatever downstream application. As John mentioned, we validated this on hundreds of different methods. And let's face it, everyone's working with finite budgets. So this is a method that will let you leverage economies of scale, batch processing, and give you the flexibility to hang on to a sample like the Kaiser Permanente study, who's used Origene on the West Coast. They had the funding to do a massive collection. They've collected over 110,000 samples, but they weren't really sure how they were going to process and analyze them. So they just let them sit on the shelf at room temperature for about 1.5 years. They got their second grant, this time from the Stimulus Fund, and now they're genotyping saliva samples on the Affymetrix Gene Titan platform, and everything's going very, very well. So there's an opportunity there to maximize the budget. And whether you're doing 50 samples or 200,000 samples, this solution is scalable, and it can be really modified and optimized to fit almost any kind of research paradigm. So we've presented a bit of history, a bit of data, but the best way to get familiar with the performance of our products is to generate data by your own hands in your own lab. And so we want to make that very easy. You can see a link on our web page that will make it pretty straightforward. You just fill in your coordinates, and we can send you evaluation kits free of charge to try out. If there's a particular assay that's unique to you that no one else does, at least this way you'll have a chance to generate your own data and gain that confidence with the Origene kits. So if you have any questions at all, I'll turn it back over to Shauna, and we're looking forward to hearing from you. Thanks very much.
Thanks, Aaron. We actually have several questions that have been sent in over the course of the webinar. First, and Aaron, I'll direct this first question to you. We seem to have a lot of people asking about archiving and banking Origene samples. What are our recommendations for long-term storage?
Great question. So I'll answer that in two ways. The first is I'm going to assume that you're going to purify a very small amount, and you're going to use that for whatever assay you have an immediate need for, and then you're going to try and hang on to the rest of the sample. In that case, I would recommend aliquoting the leftover Origene DNA saliva sample into nice compact cryo vials. Starstead makes some with a really nice gasket to minimize evaporation. Just leave that unpurified, aliquoted into these cryo vials, and keep them on the shelf at room temperature. And we've got data showing that everything will be perfectly fine for at least 5 years. That real-time data is ongoing. If you expect to, just do one test with the sample and then there's a very low probability of you going back to that archived sample, then I would suggest purifying the whole thing and then storing your isolated DNA, as every biobank recommends, either at -20 or -80. So again, we have protocols to help you out with either sample preparation protocol, and both will serve you for the long term.
Okay, Aaron, that's great. Thanks so much. And I've got a question that I'm going to send your way, John. We also have had a couple of people asking about tracking origin samples, in particular for large studies, and what's the best way to do that?
Okay, well, another great question. Obviously, tracking and logistics are very important when it comes to linking your donors to either phenotypic data and then to the sample itself. Our kits have the ability to be barcoded up to triplicate. So I think I showed a picture earlier that showed the origin tube itself with a barcode directly on the tube, and then you can actually get accessory barcodes on the outer container that are peelable and removable, and you can then link that sample to either hard copy information or some other source, and of course then scan in the barcode ID, which is unique, to some sort of LIMS system. Of course, it's quite flexible. We have our own barcodes that we provide, but we've worked with others to accommodate specific barcodes that the client requested to use, and so we can modify the system to fit pretty much whatever workflow or logistical system that someone has. And so it's quite an open platform in that regard.
Great, thanks, John. And we're running out of time, so thank you everyone for joining us today. Very shortly you will receive an email that details how you can order your Origin DNA kits for evaluation purposes and try them out for yourself. It will also tell you how you can view a recording of this webinar. Thank you again, everyone, and enjoy the rest of your week.